This is Kathy Chute from Harvard IACS, and welcome back to the last part of our day for the Women in Data Science Conference in Cambridge. Our next steps are actually to take a look at some lightning talks.
Today, I will be talking about designing reinforcement learning algorithms for digital interventions pre-implementation guidelines. There's growing interest in using reinforcement learning, RL, to optimize the delivery of digital intervention messages in areas such as mobile health and education. However, designing RL algorithms in these settings is not trivial. Unlike some areas such as games and robotics, these state transitions, actions, and rewards are not given and need to be thoughtfully constructed by an interdisciplinary team. In addition, this RL algorithm needs to run stably online across potential non-stationary or non-Markovian environments. They must be computationally feasible and handle update and decision times. They must be developed within a given engineering budget and time constraint, and the collected data must be usable for after-study analyses. In order to address these challenges, we provide a framework for designing RL algorithms for digital interventions and walk through the design of the algorithm through a case study on an oral health clinical trial. Our approach first develops a simulation environment with variants, where each variant is a test bed that encodes concerns of the scientific team. And then we test different RL algorithm candidates across these variants using our framework. And we finally want to select the algorithm that performs well across multiple variants. For example, one design decision is whether to cluster users together and learn an algorithm per cluster or learn one algorithm per user. We noticed through our experiments that cluster size 1 algorithms may have higher average reward, but clusters of size 5 algorithms achieve higher average 25th percentile rewards. Our hypothesis is that for users who do not benefit much, the algorithm may not visit enough states to learn well. But clustering allows for leveraging other users' data, which might explain the higher rewards. This is an example of a trade-off that we need to consider when making the final decision. And we will explore this trade-off and other design decisions further in a full paper.
Hi, I'm Athena and I'm a postdoc at the CC Lab in ETH Zurich. I'm very excited to present our work on segmentation of neuron cells in whole mouse brains, where in collaboration with the Institute of Neuropathology at the University Hospital of Zurich, we want to quantify the distribution of neuron cells inside mouse brains in order to study neurodegeneration and help advance the current state-of-the-art in potential therapeutic drugs. To do that, we developed a computational pipeline targeting large, centimeter-sized samples scanned with light sheet microscopy at a pixel resolution of a few micrometers. Our pipeline includes tile stitching for the reconstruction of whole brain data from the microscope scans, neuron cell segmentation using image restoration, object detection through edge detection filters, and object classification in order to eliminate potential artifacts. Lastly, we use spatial mapping of the detected cells to standardized mouse brain geometry, in this way enabling the comparison of cell distribution among multiple samples and comparison between different cohorts. By computing the average cell distribution in each cohort of interest, our pipeline has made possible the analysis of a novel drug delivery carrier by quantifying its spread in whole mouse brains and comparing it to existing delivery systems and a group of control samples. This work will be published soon in collaboration with the research team who developed the novel drug delivery system. Thank you very much for this opportunity to present our work.
Hi everyone, today I'll be presenting work that uses network analysis of job transitions to inform career advice. So while career advice has always been important, it's become particularly relevant recently as many workers were displaced from their jobs during the pandemic. And in fact, there are a number of tools that suggest job transitions to workers to higher wage employment based on job requirement similarity, but these tools do not always consider whether a transition will be beneficial for the worker in the long run. With this in mind, we hope to leverage network analysis of US resume data to suggest transitions that are not only feasible, but are also associated with living wages and stability beyond that single transition. We start by converting data from longitudinal resumes to a network structure where nodes are jobs from a list of standardized professions and weighted directed edges represent the number of people transitioning between these jobs. Once we have this configuration, we can use it to create subnetworks for each job that capture the most likely career path available from that job. We do this by grabbing all directed simple paths that start at job J and further simplifying our models by removing edges with weights below a certain transition probability, as well as terminating paths that exceed the number of transitions that could be realistically achieved within a single career. Once we have these models, we can use them to develop indicators that evaluate occupational mobility. With respect to low-wage occupations or occupations that are projected to decline, we find that the most useful indicator is the probability over the whole subnetwork that a worker will reach a job with a living wage that is not projected to decline from that job. Overall, we find that the indicators derived from the resumes and the indicators that we derive from the network models are highly correlated, providing some support for our approach. However, we also find that low wage or shrinking occupations offer limited access to stable high-wage employment. Still, there are a number of jobs that can provide workers in these professions with dependable access to sustainable employment down the line, and I hope to discuss this with you further at the poster section.
Hello, everyone. Today, I'm excited to present to you my work on personalized disease onset prediction using medical claims data. In this work, we hope to leverage medical insurance claims data to predict the onset risk of chronic conditions in order to delay and prevent the diagnosis. In the US, medical data are often decentralized, distributed among different hospitals with different formats. It is very hard to find aggregated data at a large scale. On the other hand, insurance claims data are more centralized and standardized. Therefore, they offer an unique opportunity to tackle our goal in mind, given the large quantity of observation data up to a few years of time horizon. For this work, we use proprietary medical claims data aggregated from private companies' insurance claims, consisting of more than 60,000 unique individuals of 1 to 7 years of time series data. Our methodology consists of 3 steps: data processing, feature generation, and model training, where we used gradient boosted trees as downstream machine learning model. We have conducted experiments to predict the probability of diagnosis in the next year for 50 major diseases. In this table, we show 3 diseases which we consider important and with promising results, including type 2 diabetes, hypertension, and osteoarthritis of the knee. For those, we are obtaining close to 0.8 AUC score. This is still an ongoing work, and we're very happy to receive feedbacks and suggestions. Thank you very much for your time and attention.
Hi, my name is Helen Landor, and I'm excited to present to you today about my research regarding the tactics, techniques, and procedures, or TTP, of disinformation related to climate change in the European Union. For this project, we use data from EU versus disinfo.eu. As shown on the left, the data is publicly available on the website, and it provides a summary, disproof, and link to a full article of disinformation. We took the full article, auto-translated it, and then ran the summary, disproof, and full text through sentiment analysis and argumentation analysis in order to examine the tactics, techniques, and procedures of disinformation. First, these are the results from the sentiment analysis using Natural Language Toolkit in Python, or NLTK. As you can see, the summaries and disproofs have some neutral sentiment. However, the translated full articles are extremely polar. This finding agrees with some current analysis of disinformation that argues that most disinformation is highly polarizing. Next, we use the PRTA model to extract the literary techniques of persuasion used in the disinformation, as well as the summary and disproof. As you can see here, there's an example of highlighted text, as well as a couple snippets pulled out by the PRTA model of the climate change disinformation related texts. When the results from PRTA are normalized and aggregated, they look like this. Using each bar, you can compare how much of the labeled spans belong to each category of technique in the chart below. By examining this, we can see that there is great variation in the technique used by news analysts in the summary and disproofs, as well as that used by the original author in the full text of disinformation. Thank you so much for listening to my talk. I look forward to feedback.
Hello, my name is Jennifer, and I'm going to talk about predicting Clostridius difficile recurrence from high-dimensional microbiome data. Clostridius difficile infection, or CDI, is a debilitating and potentially deadly disease with a high risk of recurrence. After recurrence, the risk of subsequent recurrences continues to increase. At this point, research into factors that cause individuals to recur is limited, and there's no reliable way to predict CDI recurrence after initial infection. To this end, we conducted a study that collected fecal samples from participants diagnosed with CDI before treatment, after treatment, and for 4 weeks or until recurrence. All samples were analyzed to determine the unique microbes and metabolites present in the gut. This resulted in a data set with 2,509 unique microbial sequences, or ASVs, 1,387 metabolites, and 9 targeted short-chain fatty acids. After filtering and transforming the data, we performed univariate analysis to determine which microbes or metabolites were significantly associated with future recurrence before antibiotic treatment, 1 week after treatment, and 2 weeks after treatment. After correction for false discovery, we found many ASVs to be significantly more abundant in non-recurrers at each time point, and several metabolites to be significantly associated with outcome at week 2. However, when we used predictive analysis, we found that only untargeted metabolites were predictive of recurrence, achieving AUCs of 0.77 at week 1 and week 2 after initial infection. We identified metabolites found to be associated with outcome in univariate and predictive analysis and found that many metabolites were the same across methods. Moreover, the metabolites fell into 3 groups that may begin to explain the different metabolic state between recurrers and non-recurrers. For example, we found that glucuronidated compounds were higher in recurrers, which may indicate decreased microbial deconjugation activity. We also found higher levels of anti-inflammatory endocannabinoids in non-recurrers, while recurrers had higher levels of metabolites like sphingomyelins, indicating inflammation and cell death.
My name is Michelle Lee, a PhD candidate at Harvard, and I am super excited to share our work on deep contextual learners for protein networks with you all. This work is motivated by the decades worth of biomedical discoveries enabled by modeling interactions between proteins. In fact, protein interaction networks have been crucial for disentangling the rich diversity of complex biological phenomena. However, these networks are typically presented as generic maps without any contextual information about the particular cell type or tissue that the proteins are interacting in. Although cell type-specific PPI networks can model contextualized interactions at the cellular level, on their own, they cannot generate optimized representations and provide predictions in downstream tasks. Deep learning models designed for graphs can help, but even state-of-the-art models cannot automatically learn information like cellular and tissue context for protein interaction networks. Rather, we need domain knowledge to infuse this information into the graph neural network architecture. So we propose AWARE, a multi-scale graph neural network that integrates cellular and tissue contexts into the architecture. The aim is that with our context-aware embeddings, we can make better downstream predictions across biomedical applications. First, we construct our cell type-specific PPI networks and cellular and tissue hierarchy, and apply our model that is designed to learn the topology of proteins, cell types, and tissues to capture the behavior of proteins across cell types and tissues. As a result, our method is able to learn a unified multi-scale embedding space for proteins, cell types, and tissues that capture multi-scale biological organization. Excitingly, these embeddings are able to capture generic PPI embeddings in predicting cell type-specific disease gene associations. We envision our method to open up new possibilities for contextually adaptive embeddings in biomedicine. Thank you for your time.
Hello, everyone. Thank you for an opportunity to present computer vision transfer learning applications at Women in Data Science Cambridge 2022 conference. My name is Mina, and I'm a manager of marketing science at Computational Group. This presentation is inspired from my mentoring experiences at various MIT hackathons. The purpose is to apply AI for the benefits of humanity. This presentation will briefly walk through how to leverage transfer learning to better detect and classify diabetic retinopathy. It is one of the leading causes of vision loss worldwide. For both training and testing process in this presentation, we are going to use retina images taken under various conditions that involve noise. There are different types of classes, 5 classes labeled from 0 to 4 based on the severity of the disease. This slide highlights the model training process. We are going to use efficient V3 as the pre-trained model to improve the speed and performance efficiency. The next few steps will highlight the hybrid approach. So rather than directly applying the entire pre-trained model, which will not necessarily improve the result, we are going to freeze the first few layers and add new trainable layers on top of the frozen layers for future learning and extraction. The next slide highlights the layers and parameters that are going to be added to the efficient V3 model. So leveraging the benefits of transfer learning combined with the customized architecture will help improve the speed and performance efficiency at higher robustness and lower bias. This approach can be applied to different types of imaging problems to save time and resources and generate more reliable outcomes. And this concludes the presentation. Thank you.
Hello, everyone. My name is Yan Zhang. I'm here to talk about a new feature we recently added into the Recommenders repository, an open source repository that includes state-of-the-art recommender algorithms and the best practice of applying them. Recommender systems are built into many applications in our life. They aim to predict users' interests and recommend product items that users are likely to engage with, such as to click, purchase, or to spend longer time watching, reading, listening, etc. When evaluating a recommender system, accuracy metrics are commonly used. However, when solely focusing on accuracy metrics, the items being recommended always tend to be popular items, which are the items mostly sold or viewed. It leaves the long tail less popular items having less chance to get introduced to the users. We argue that the ability of recommending long tail items should be taken into account when evaluating a recommender system in certain scenarios. To evaluate how well a recommender can recommend long tail items, metrics such as coverage, novelty, diversity, and serendipity are good candidates to use. The major contribution of this work is that we have implemented these diversity evaluation metrics in the recommender repository in PySpark and Python. Why it matters to include diversity metrics? We summarize our thoughts from these four aspects. In an experiment of comparing an ALS recommender and a random recommender on MovieLens 10K data, we show following comparison results. It shows that the ALS recommender performs better on ranking metrics, while the random recommender performs better on diversity metrics. This is because ALS is optimized for estimating the item rating as accurate as possible. Therefore, it performs well on accuracy metrics, including rating and ranking metrics. Usually, there is a trade-off between one type of metric versus another. One should decide which set of metrics to optimize based on business scenarios. Thanks for watching. Please feel free to check out sample code from the Recommenders GitHub repository.